This lecture is, again, on the digestive system, in this case on the organ called the pancreas, which is another of your accessory organs. The liver, which you've already heard about, is an accessory organ as well. Now we're talking about the pancreas. So the pancreas has two roles, and one of them is its endocrine role or endocrine function. So this is the secretion of hormones. For example, the hormone insulin. But that's not what we're going to be talking about today. That has to do with your endocrine system, and we'll discuss that when we're talking about the endocrine system. What I'm going to be talking about today instead are the exocrine functions of the pancreas. Now remember, when you have a gland secretes substances. That's all it really does. It's made of glandular epithelium. There are two types of glands. There are exocrine glands and there are endocrine glands. Exocrine glands are the ones that secrete their substances into a tube, usually called a duct, but basically a tube, a garden hose, if you will. And the secretion then flows through that tube to its intended target, to where it's supposed to wind up. On the other hand, endocrine secretions from endocrine glands simply go into your bloodstream and are carried all over the body, and they affect their target tissues by way of some fancy chemistry, which we will discuss another time. Right now we're talking about the exocrine function of the pancreas, which in this case means that various pancreatic secretions flow through a tube to the place that they're going to wind up affecting. So what happens here is that the pancreas then secretes water, electrolytes. Electrolytes are basically minerals, things like sodium and potassium and the like. And digestive enzymes into the small intestine. So the pancreas then secretes these substances. They flow through a tube into the small intestine. So what happens then is that the fluids and the electrolytes then dilute and buffer the acids in chyme. Remember, chyme is flowing out of the stomach into the small intestine. It is highly acidic because it's got hydrochloric acid from the stomach in it. And the water, the fluids, will simply serve to dilute the chyme, so therefore dilute the acids. But then we also have these electrolytes that buffer the acid. A buffer basically is a chemical that will take a strong acid or a strong base and make it into a weaker acid or base. So here we have the acid level, the pH level, being brought more nearly to neutral by the action of these things. Okay, So that then has to do with the electrolytes. And then we have the pancreatic enzymes. And these do most of the digestive work. Then, in the small intestine. So these then break down ingested materials into absorbable units. Absorbable units means units, basically small molecules, that can pass out of the small intestine through the lining of the GI tract into your bloodstream. So these are going to be small things. These are going to be small sugars. These are going to be amino acids. These are going to be salts. These are going to be fatty acids. So the component molecules of these things that make up your food, the food has to be broken all the way down. Remember, digestion technically means the chemical breakdown of these food molecules into really small molecules, which can be absorbed. So those are then the absorbable units. Okay, so let's take a look at these enzymes. They come in different classes, and the names tell you their job, so it's kind of nice. So then these enzymes, there are different categories of them, and they include lipases. Okay, now when you see the suffix ase, that basically means breaks down. So lipases, no surprise here, attack and break down lipids. Remember, lipids are your fats and oils. Then we have carbohydrates. I think you can figure out what that does. So these then digest sugars and starches. That is to say, they digest carbohydrates. So they break carbohydrates down into really small sugar units that can then be absorbed. And then we have proteolytic enzymes. Okay, and these actually come in two different varieties. Now, where you see lit or lyse or anything like that, that also then means breaks down. So when you see lysis or anything like that, it means breaking down. So here we have proteolytic enzymes. We have two different kinds. So your proteolytic enzymes then break down proteins. But proteins are huge molecules, so we actually have two different classes of proteolytic enzymes in order to take care of these. First, we have what are called protein aces. So these then break down proteins, and these then break down large protein molecules. And once you have broken large protein molecules down into small protein molecules, they're still not small enough. So then we have what are called peptidases, because small proteins can be known as peptides or polypeptides. And these then break down small peptide chains. And that is it.